Welcome back. We're starting a new organ system, the lymphatic system, also known as the immune system. So I'm going to call it the lymphatic and immune system. And the first thing that we are going to talk about before we get into the nitty gritty of either what is involved in your lymph system and what is involved in immunity is we're going to talk about microbes and pathogens because that's what this organ system is going to be fighting. So when we're talking about microbes, we're talking about microscopic organisms, okay? Microscopic things, bacteria, viruses, and something you may not know anything about, things called prions, which I'll explain to you in a few minutes. So if it causes diseases in humans, it's called a pathogens, okay? So not all microbes are pathogens. Okay, but we have some pathogens which are living cells, such as parasites or protozoa or fungi, like athlete's foot. Okay, bacteria, and some that are not alive, that are not cells, and these are our prions and our viruses. So examples of some microbes that you may know about causing diseases would be here in the upper left, this cute little organisms with a really long flagellum, Vibrio cholera. This is what causes cholera, which is very common source of severe disease causing profound watery diarrhea, which can kill children in a day or two in areas like after natural disasters and places of warfare where people are crowded into massive amounts of people into small areas. And the lower left corner, this is an organism, Escherichia coli. You've heard it called E. coli, a very common bacterial disease. The lower right corner, this is Streptococcus, which is what gives you strep throat. And this cute little blue spiral organism in the upper right corner, Treponema pallidum, this is what gives you syphilis. So let's try not to get that one either. All right. So normally we've got like 3 different lines of defense. Our first line of defense are going to be our physical and chemical barriers. In other words, let's keep this stuff out of our body. But if these barriers are broken and these microbes get into our body, our early responses are going to be non-specific. So think of this like the defensive line on a football team. It doesn't matter who's on the other side. It doesn't matter who they're playing. They're going to be trying to prevent that team from scoring. Okay. And and so yes, I know that there's individual different players. And yes, I know there's individual different plays that you can call. So there are different things that we can do with this non-specific response. We can talk about calling different white blood cells to do phagocytosis. We're going to have inflammatory response, complement, interferon. I'll be going through these in detail much later. And then if this fails, then we're going to move to a specific response, which means our B cells are going to get involved and try to knock it out, and our T cells are going to work in a totally different way to try to knock it out. And they're going to be very specific with what they do for one organism only. All right, so let's get back and talk about bacteria first. So bacteria are single cell organisms, and they belong to that big group of organisms known as prokaryotes. Okay, remember, kary means nucleus, pro means before. So bacteria do not have a nucleus, as opposed to humans, which are eukaryotes, meaning we have a true nucleus. And bacteria are very small. So this is not a pencil tip, this is a pin tip. And as you can see, if we took that E. coli organism and we put a bunch on the tip of a pin, you would see how tiny they are in comparison to the tip of a pin. So no, you cannot see them with your naked eyeball. And they come in three common shapes. So that rod shape is called a bacillus. You don't have to know the official names of the shapes. I'm just going to say them in case you've ever heard them before. So E. coli is a bacillus. We also have round shapes, which are a coccus, like streptococcus, the strep throat organisms. And then the little spiral or the little curved ones, like in syphilis, that's called a spirillum. So let's look at the anatomy of a sample bacterium, and in this case, yes, it's a rod-shaped one. All right, so the first thing we're going to notice here, the yellow is the plasma membrane. And so just like human cells, they have a plasma membrane, but now they have something that eukaryotes don't have. Outside the plasma membrane, they have an actual cell wall. Okay, so that's why I said you could not call the plasma membrane the cell wall. They are two distinctly different things. And the cell wall is much more rigid. And then some bacteria outside the cell wall have a capsule that is gelatinous and is sticky. And what this allows that bacterium, when it's on a surface, so say like thinking something that's sloped or something that's straight up and down, allows it to stick to it. So like your cell phone, if you're in the bathroom and you have touched it before you've washed your hands, because you know a lot of people touch their cell phones while they're sitting on the toilet, not a good idea, because you've touched your cell phone again before you've washed your hands, and so you have organisms on there. Okay, and then for those of you who do such a good job of washing your hands before you leave the bathroom, do you hold on to that paper towel to open that bathroom door? Or do you throw it out and then use your clean hands to open the bathroom door? Forgetting the fact that people who didn't wash their hands have touched that bathroom door with the organisms on their hands. And so by taking your clean hand and touching it, you now have organisms all over your hands. Okay. So don't do that. All right, getting back to the anatomy of a bacterium. We left off at the capsule. Most bacterium are going to have a flagella on one end, and the flagella, instead of moving like a snake, they actually rotate. And when they rotate, that bacterium moves backwards. So here's an example of that, um, like that Vibrio cholera organism. And as you can see, it has multiple flagellum on one end, so it can really go fast backwards. Some bacteria will have flagella on both ends, so they can go this way or they can go that way, depending which flagella are moving. Okay. Notice there is DNA inside this bacteria, but there's no nucleus. There's no nuclear membrane, no nuclear pore. There also is this little round circle right here, which is nucleic acid. I'm not going to tell you what that is, but I want to let you know that there is this other little thing. And the reason for this is actually the part of the bacterium which gives it antibiotic resistance. But I'm not going to ask you about that in this course. But before you send me a message and ask me what that little circle is, now I've told you what that little circle is. Not that we have to know it for this course. Continuing on, you can see these long, skinny things, each of which is called a pilus, and these are hollow things, and there's three of them actually here on this photomicrograph of a bacillus. And what they do is they allow this bacillus to send DNA to other bacteria so they can transfer their antibiotic resistance to other bacteria. Kind of a cool thing. All right, so how do bacteria reproduce? Because there aren't male and female bacteria, there's only the one bacteria. So this is showing, okay, so the DNA of this bacteria. So in reality, it doesn't really attach to the inside of the plasma membrane. It's just showing you there so you can keep track of how many there are. So yes, the chromosome is going to duplicate, as well as all the intracellular contents are going to duplicate. And then once the cell has duplicated anything, it's going to start dividing. And once cytokinesis is over, you are going to have two daughter cells. And so this bacterial reproduction is known as binary fission. Fission is division, and binary means two. So you're resulting in two cells, each of which are identical to the original cell, and each of which are identical to each other. Now, if a bacterial cell lived in perfect conditions, good nutrition, the right amount of moisture, everything, the right temperature, it could reproduce every 20 minutes. So let's assume you gave it the ideal conditions. Okay, like you were wanting to culture the bacteria for something. If you put one bacterial cell in it, how many times would it undergo binary fission? How many cells would you end up with if you returned 24 hours later? In just 24 hours, that one organism would end up with almost 70 trillion organisms. So think about that the next time you don't wash your hands. Okay, so here is a picture of one bacterial cell undergoing cytokinesis. So, in some respects, you could say that this is asexual pornography. Okay, moving on. So, bacteria, as we know, can cause diseases, and they can cause diseases in a couple of different ways. And one way they cause disease is just by the numbers of bacteria growing, causing diseases, and they're going to be feeding on the normal tissues. And so, an example of bacteria that does that is a normal bacteria that we find on the skin of everybody, and that is staph. Specifically, it's known as Staphylococcus aureus. So, staph. And so, a little staph infection of the skin often starts as a little tiny little pimple. And it's got a little redness and swelling around it. And I'm sure everybody has had one of those. And instead of putting a warm compress on it, which is, like what your mother or your healthcare provider tells you what to do, most of us want to go there in the bathroom and try to pop that sucker. Okay, and what happens is when you squeeze it, you are sending those germs deeper into your tissues. And then what happens is you have a worse infection. Okay, so what is all this white and yellow stuff? This is pus, which basically is showing that your body is trying to fight this infection because those are dead cells. They're dead tissue cells and dead white blood cells. The redness shows that you have an inflammatory reaction. So that is good. If you didn't have any redness and pus, that would be bad because then you would be dying of this. But what happens is that this staph infection, as it takes hold, you're going to end up with like a little ulcer. If you could clean out all the pus and everything else, you would end up with an ulcer like this. And if you could keep it clean and keep the staph out of it, eventually this ulcer would start shrinking down about a millimeter a day, and you would end up with a not pleasant looking scar, to put it mildly. Now, the problem with a lot of staph infections is that they are resistant to a lot of antibiotics, and probably a lot of you have heard about MRSA, which are methicillin-resistant Staphylococcus aureus infections, which is a big problem in folks who are hospitalized or in group homes or in old folks' homes or any of those group care facilities. Now, another way that bacteria can cause diseases is that some of them will release a toxin, and a toxin is a molecule that is going to screw up some aspect of metabolism that's normal for your cells. Okay, it's a little protein that is playing with your cellular metabolism. Now, the good thing is that our bodies will recognize this toxin as an foreign, and so we can mount an immune response against it. But if it doesn't, here's what's going to happen. And here's an example of a bacteria that does it. Have you ever been told, don't go buy those dinged cans in the back of the grocery store where they're cheap? or if you happen to drop a can that you bought at the grocery store, like open it and try to eat that right away. Don't let it sit on the shelf, because when you get a little dent in a can, what happens is you have little microscopic cracks, and you are now letting oxygen inside that can. And that can was sealed so no oxygen could get into it. And that was the whole point of canning. So organisms that inside that required oxygen to grow could not grow. But now, because of the dents, now they're getting oxygen. And an example of an organism that can grow is Clostridium botulinum, which gives you botulism. Okay. And one of the things that this organism does is it causes contraction of skeletal muscles, and it doesn't allow um, the muscles to relax. And so this is going to happen with big muscles. So imagine you could take a breath, which is muscle contractions, but you couldn't exhale, and take a breath, but you couldn't exhale, and take a breath, but you couldn't exhale. And so here you can see this drawing from way back in the 1800s, because I didn't want to put actual photographs of people with this disease. But basically, the way this kills you is you end up suffocating to death because your muscles don't relax, so you cannot inhale. Not a good way to die. Okay, so you know how they always say, take your food and be and plop it in your refrigerator. And everyone goes, no, 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 we need to let it cool down before it goes in the refrigerator. And it's like, no, you can't let it cool down. So does refrigeration kill the bacteria? Well, and an answer, no, absolutely not. Refrigeration does not kill bacteria. Okay. But how fast those bacteria reproduce depends on moisture content and the temperatures. And so if we look at the temperatures of refrigerators and freezers, bacteria growth is slowed, but it is not stopped, which means as soon as you take that food out and you put it on the counter, what's going to happen? Bacterial growth is going to start increasing. So my mother-in-law used to love to take meat out of the refrigerator. Like first thing in the morning, send the kids to school, take the meat, put it on the refrigerator, remind her all day what she was cooking. Like, you can't do that. You can't let it sit at room temperature for 8 hours before you cook it, because the bacteria are growing. Okay. And this is going to give you food related diseases unless you kill it by high temperatures, like boiling or thorough cooking. Well, granted, when she cooked beef, she always cooked it well done. So yes, everybody ate shoe leather because that prevented having bacterial diseases from, and she could let it sit out all day. But I'm going like, just let it sit in the fridge and take it out before. So here's the thing. If you have a piece of meat that came from the butcher's, okay, like a steak, you got to realize that the surface of the steak is contaminated with bacteria. It's contaminated from when it was slaughtered. Why? Because there's poop in a slaughterhouse. There's no other nice way to sell it. There's fecal material all over the surfaces. So it's on the surfaces of the meat. Okay. Then you have those big pieces of meat that went to your grocery store, and then they chopped it up into smaller pieces. And it doesn't matter they were wearing gloves, because they had knives that cut through the outer surface of that piece of meat and dragged those bacteria to the inner surface where they were slicing it. So now there is meat on there are fecal bacteria on the outer surfaces of that very expensive steak that you bought. But here's the good news. When you cook that steak, you're going to be searing the outside. So even if it's medium rare on the inside, you have seared the outside. So if you have kept that steak in a refrigerator until the time when you seared it, okay, you can leave it outside for 20 minutes before you cook it, as long as you have a good sear and you can see that brownness coming in to the meat, you're going to be okay. But what happens when you take that piece of meat and you chop it up to make steak tartare or to make hamburger beef? Well, now you've taken the pieces of the meat that have all the bacteria and you've mixed them up with the inside of the meat, which didn't have the bacteria. And now you've made this juicy, beautiful burger. And this is the kind of burger most of you want to eat because it's pink and it's got the juices. And now you're just salivating, saying, "Oh, Dr. Pal, that's what I want for dinner tonight." And I'm going, "Please don't. Please, please don't." Okay. You cannot eat hamburger because the bacteria that looks like this, because the bacteria on the inside are not going to be killed until they reach at least 140 degrees. And that is not going to happen if the meat is pink. Take out your thermometer and test this. So when you're cooking burgers, it's not until it's medium well that you're going to have your burger meat over that 140-degree temperature. Don't even ask for medium, because when you ask for medium, 90% of the time they give you a medium rare part right in the middle of it, which defeats the whole point of it. Okay. And like if you're going to a fast food place, if there is in that burger, take it back. Take it back. Okay. Because across this country, almost every month, there are dozens of people that get sick with E. coli at a fast food place. It's not just McDonald's. You pick it, you're going to get sick from it. Because folks want pink in their burgers for some godforsaken reason. All right, let's talk about viruses. So here are some beautiful little viruses. Here's the influenza virus cruising along the little cilia of your tracheas, so you can sweep them along. Here's your coronavirus, COVID-19. Most of you can recognize this one with its little protrusions on the surface. It looks kind of similar to HIV at first glance, but in reality, they're not similar at all, because HIV is an RNA virus for starters, totally different. Up here in green with these really long surface projection, this is a common cold virus. And this one over here that looks like flowers, here's your polio virus. So they're all kind of cutesy, aren't they? But no, they're nasty. So here's the thing about viruses. None of these are cells, which means none of these are alive. And yet they can make you sick. So we're going to learn how this happens. But before we do this, let's talk more about what is a virus. Well, at most basic, a virus is a nucleic acid inside a protein coat. Okay, so you weren't looking at a nucleic acid inside a protein coat in any of those pictures I just showed, because those viruses have a whole bunch more stuff. Why? Because they want to get inside your cells to make you sick. So what I mean by nucleic acid inside a protein coat is a virus must have a protein, what we call capsid. This is the coat. It's called a capsid. And inside that outer capsid, there is going to be an inner core, which is a nucleic acid. And depending on the virus, it can be DNA or it can be an RNA virus. Okay. And this DNA RNA contains the genes that this virus needs to replicate itself. So if it's a DNA virus, then the DNA can actually go into your host, your cell DNA. And now it has the genes to replicate itself. If it's an RNA virus, when it enters your cells, it has the enzymes and the directions to take that viral RNA and change it to viral DNA. And then that viral DNA will go into your cell's DNA. Now, so what does that mean? That means in addition to the required nucleic acid and the required protein capsule, that almost every virus is going to contain enzymes which are going to help it reproduce inside the cells. So a typical virus looks more like this. And do you see how this looks more like that coronavirus or that HIV virus or that polio virus? So right here in the middle, there's our nucleic acid. This one's DNA. And then this blue thing, that is the protein capsid. And then here on the outside, this is called the envelope, and it's got like these spiky things. And these spikies are receptors that can bind to different cells in your body. And where it can bind to receptors in your body, those are the cells it can get into where it's going to create havoc and make you sick. So, can it get into cells in your brain? Can it get into cells in your lung? Can it get into cells in your GI tract? You can get into cells in your GI tract, you're going to get diarrhea. You can get to cells into your lungs, you're going to end up with pneumonia and trouble breathing. Okay. Now, in addition to that kind of typical virus, there's this one which looks totally different, looks like a little bug. This is called a bacteriophage because this is the type of virus that infects bacteria. It's a little bit different. And yet, right up here at the top, there's your nucleic acid inside the capsid. And then it's got this little thing right here, which is where it injects it inside the bacteria. All right. And now, even though I made these look the same size, this one is much smaller than this one. You can see because this part right here is like this side. This one's much tinier, this little spidery bacteriophage. So if we compare sizes here, and the reason why I showed you bacteriophage is because that's what they're using for their example of the virus. In this picture, you can see how tiny this virus is compared to our bacterium here with the little flagellum. Think our cholera bacterium. And you can see how small that is compared to a normal cell with some endoplasmic reticulum and a nucleus. So the largest virus is only 25% the size of the bacterium and only 1% the size of eukaryote. So if we look down here at around 10 nanometers, that is about the size of the poliovirus. So it is about 5 times the size of a protein molecule. And then about 10 times larger than that is going to be the flu virus. Okay. And then about 5 times larger than that is going to be a smallpox virus. And about 5 times larger than that is going to be a bacteria. And then 10 times larger than that would be here, and 15 times larger than that, that would be your animal cell. Okay. So outside of a cell, a virus is just like having chemicals sitting on a shelf. As long as they're sitting up there on a shelf, it's self-contained, it can do you no harm. But once it's inside a cell, it can replicate. It appears like it's alive, but it's just a cellular parasite. So let's see what happens once it's inside your cell. Think of it kind of like a microscopic parasite. So like Captain Jack, what does he do? Well, they commit a lot of crimes, don't they? So a little breaking and entering into the cell. Then it hijacks the cell and tells the cell, do what I want you to do. And what is it I want you to do? Well, pirates are a little narcissistic. And what they want you to do is to make a lot of them. Just make a lot of them. Okay? And once you've made thousands of copies of them, then they're going to escape to break and enter into other cells. So what does this look like in a scientific drawing? Up here. All right. So here is a very simplified virus, okay, showing that it's going to attach to the surface of the cell, that it's going to drop its contents into the cell. All right. And so the purple little squirrely things, that's its viral nucleic acid, which is going to, and because it's single stranded, it's showing that it's RNA. So that's going to enter the nucleus of the cell. It's It's going to become viral. DNA is going to enter the host cell's DNA. We're going to have transcription. We're going to make messenger RNA. We're going to leave the host cell's um, nucleus. We're going to come over here. We're going to have translation. Now we're going to be making a bunch of viral RNA. We're also going to be coding for and making all the other parts of this virus. So we're going to making its protein coat. If it had enzymes, we're making its enzymes. If it had spikes on its envelope, we're making all of that. And then what's going to happen, this is all going to be repackaged together, and then it's going to bud and escape. So that's viral reproduction at its simplest. Okay, so now let's move on to the third thing, and those are prion proteins. And prions basically are proteins, and we know we need them, but we don't really know what they do in healthy folks. But we know they're on the surface of many cells, especially your neurons. So we've got lots of them in our brains, okay? And we do know that they're required to have our synapses functioning normally. But how they do that, don't know. Okay. So here's an example what a prion is. Do you see how it's just a folded protein and like some of it is a helix and some of it is pleated? And if this is what a normal prion would look like, what happens is prions can go rogue and they change their shape. So this same prion could be folded like this, where you can see it's got a lot more pleating going on. And that's called the rogue form. And you can see if the structure has changed, the function must change. So in other words, this is not going to do whatever the job was it's supposed to be doing. It's not going to be able to do it. Okay. So the rogue shape prion doesn't function. But one thing a rogue prion can do, if it's sitting next to normal prions, it's like giving an invite. Hey, come join the party. And it converts normal prions into the rogue form. And these rogue prions cause degenerative diseases in our nervous system. Now we know there's a latent period until these degenerative diseases become apparent. And then from the time they are apparent, from the time of death, can be quite short. It could be a few months to up to a couple of years only, but it's quite short. So the good news is these diseases are rare. So one example of disease is something called Kuru. I'm sure you've never heard of it because it happened in tribes in Papua New Guinea who used to do cannibalism. Yeah, didn't hit the press a lot because, that went away more than 100 years ago, or about 100 years ago. Creutzfeldt-Jakob disease. We got less than 1,000 cases here in the United States. Where we had a lot more cases was in Japan. Okay. Bovine spongiform encephalopathy. Ever heard of that? Probably not. How about scrapie? Scrapie is a disease in sheep and goats. Cannot be sent to humans. It's called scrapie because the sheep and goat take their hooves and they start scraping their fur off to the point where they are scraping and making ulcers in their skin. Okay, but bovine spongiform encephalopathy, that can be transmitted to humans. And this is what the brain looks like. And as you can see, it's not normal. You have lost a lot of your white matter. Okay. So if this is what normal white matter looks like with a lot of normal healthy neurons, here's what it looks like in a spongiform encephalopathy. Spongiform just means it has the form, the appearance of a sponge. As in, see all this space? Does it look like your kitchen sponge? Yes. All right. And I'm actually 100% certain you have heard of bovine spongiform encephalopathy because that is just the name for mad cow disease. Okay, so if you eat the meat of cows with mad cow disease, you can get bovine mungiform encephalopathy. Okay, Kuru, Creutzfeldt-Jakob disease, that's because they're eating the brains, because it's in the brains where the prions are. Okay, but it used to be when cows had this disease, they could slaughter the cow and they could use these slaughtered cows as food for other cows. Okay, and so that shows where the location of prion diseases were. So the Creutzfeldt-Jakob disease was over here, Kuru was over there. And then mad cow disease was over here, and the bright green was where slaughtering and feeding slaughtered cows to other cows was outlawed first. And now the rest of Europe has outlawed it, and where it is outlawed in North America kind of depends what part of North America you live in. So we'll just kind of leave it at that for right now. Okay, so thank you so much. We're done at this section. We'll come back and we will actually start learning about the lymphatic system, functions, and structures. See you soon.